Okay, so now we have the presentation of Jessica Abbott from Lund University. She has been working on sexual dimorphism, and today she will be speaking about using experimental approach for studying evolution of sex chromosomes.
Yes, thank you, and thank you very much for inviting me. Here on this slide, you can see two of my study organisms. Oops, that's the one. Fruit flies, which I'm going to talk about today, and flatworms, which I'm going to mention briefly, but not really discuss in any detail. So over the past 5 to 10 years, I've been trying to use experimental evolution to test theories about sex chromosome evolution. And I'll just go through this quickly because I assume most of you understand how experimental evolution works. You have some organisms in the lab, you apply some sort of treatment to them, you let a number of generations pass, and then you look to see how their phenotypes have changed. And nowadays we can also use sequencing data to try to figure out what genes might underlie these changes. So that's what I've been doing, but I've been trying to manipulate the selection pressures on males and females and/or the sex chromosomes. Thomas already gave a little overview of sex chromosome evolution and how we typically expect it to proceed, so I'm not going to talk about that in very much detail. But the things that I've been working on have been trying to investigate different stages in this evolution. And we expect to see degeneration of non-recombining sex chromosomes. Sex chromosomes are also interesting because you can see these faster X effects, that you can have more efficient fixation of loci on the X or alleles on the X, and that you can have these sex-limited rates of evolution and sex-limited selection. So that's why I think this is a particularly interesting sort of topic to study. So one of the things that I've been working on is trying to mimic the evolution of new sex chromosomes in the lab, and this is using these hermaphroditic flatworms. I've been giving a talk about this at ESEB, so if any of you are going to Prague and are really curious to hear about that, then you'll have the opportunity in August. Another component was trying to look at the possibility for co-evolution between the sex chromosomes, so thinking that if you have degeneration of the Y, that perhaps the X can compensate for this somehow. Outside of the realm of sort of dosage compensation, and that's something that we published last year. And today I'm mainly going to talk about this female-limited X chromosome experiment that we've had going on for a number of years. And this is basically trying to look at what happens when selection pressures on a portion of the genome becomes sex-limited. So basically, what happens if the X becomes limited to a single sex, then it becomes sort of like a neo-W in a way. Yeah, so that's the one I'll discuss. Okay, so why did we pick the X chromosome to do this, and why were we interested in female-limited X chromosome evolution? Well, partially because I had done a male-limited X chromosome evolution experiment previously, so I wanted to follow up on that. But also because the X is potentially enriched for sexually antagonistic genetic variation. This is something that, when I was designing this project a number of years ago, then it seemed like there was some decent evidence for this, but now there's a number of studies that have come out over the past couple of years that suggest that this might not be the case. But luckily, there were other reasons why the X is interesting. Partially because even if it's not enriched for sexually antagonistic loci, you still might find some signature of sexual antagonism, and we can look at predictions about the dominance of these loci. And also just because it's interesting to think about whether the X should be closer to the male or the female optimum, because there's different lines of argument here that argue in different directions, basically. So you could say that because the X spends more time in females, this leads to feminization or demasculinization of the X, and there is some evidence to support this. But sexual selection is also stronger in males, and so this might make it easier to fix male benefit alleles on the X. And we also expect that selection of X-linked loci will be more efficient in males due to their heterozygosity. And so it's not really clear what the overall outcome of this should be. So we wanted to try to look at this by saying, well, what happens when we remove male-specific selection on the X? Does it become specialized for female fitness, and if so, how? Okay, so to do this in the fruit flies, we use this FM balancer. And so here, these flies, you can see that the male has this bar eye phenotype, and they both have yellow bodies, and the female has this hemibar. These are markers to indicate the genotype of the individual. So a female that carries 2 copies of this FM balancer will basically have a similar phenotype to the male in this picture. And the FM balancer is an X chromosome that contains at least one or maybe multiple inversions and stops it from recombining with a wild type X. So it contains the same gene content, basically, but it doesn't recombine is the idea. So by mating an FMY male with an FMX female, then you can cause this wild-type X chromosome to go from mother to daughter over and over again and never be expressed in males. Right, and so the selection here then becomes somewhat similar to that of a proto-W. And in this experiment, we had two controls. We had a wild-type control, which was to control for some of the aspects of the experimental protocol that were not directly related to sex-specific selection. So we had to select the females as virgins in order to be able to carry out very controlled crosses. So the wild-type controls for those sorts of things. But we also had a methodological control, the CFM treatment, as we called it. And in that treatment, what we did was we basically tried to incorporate the FM balancer, but keep the selection pressures on the X the same. So what we did is we passed the X through females 2 generations, and then in the next generation, it was in a male. So we did this systematically, 2 generations in females, 1 in males, 2 in females, 1 in males. And the idea was to be able to disentangle the effects of the balancer from the effects of the selection on the X. So our predictions look basically like this. So here you can see what we would expect to be a sort of selection effect, a female-limited selection effect, so that if the 2 controls are similar to each other and the female-limited X chromosome treatment is somehow different, then this should be a result of selection. Whereas if the 2 treatments that have the FM balancer are different from the wild-type control, then this would probably be some sort of effect of the FM balancer. And presumably not very interesting then, because it would just be a confound. This was initially what I expected to see. There are other patterns that are possible, of course. You could have some sort of effect of the FM balancer that seems to be reinforced by the female-limited selection, or you could also have various types of canceling out of an effect, so that you could have some sort of effect of the balancer that more or less seems to be canceled out by the female-specific selection. But I didn't really think this was very plausible, so I didn't spend an awful lot of time thinking about this sort of thing. I think you can gather where I'm going with this now, that we maybe found some things we didn't initially expect. So the good news is that we did find some results that were consistent with our predictions, that we should see some sort of feminization. So we did see evidence of feminization and sexual antagonism in expression. So this is comparing to a previous data set where we could see that things that were associated with male fitness and female fitness, they changed in the direction that you would expect. So things that were associated with male fitness, they were generally down-regulated in females after our experimental evolution. And things that were associated with female fitness were upregulated. And the same in males, but the sexual antagonism parameter, basically if it's positive, it means that it's good for males and bad for females, and if it's negative, it means that it's good for females and bad for males. So this is consistent with change in expression of genes that were good for females and bad for males. We also found significant overlap with genes related to the intensity of sexual conflict, which was kind of interesting, maybe not something that we initially expected, but of course sex-specific selection pressures are going to be related to sexual conflict, so it's perhaps not that strange. We also saw a change in body size, which is consistent with sexual dimorphism. We measured this a couple of times. This is one of the earlier measurements, but here in black we have the wild type, in green the control FM, that is the methodological control, and then in red the FLX. And so we can see in both sexes, particularly the FLX flies have gotten bigger. And because females are bigger than males in Drosophila, then this would be what we would expect under feminization. In terms of the predictions when it comes to dominance, when we're looking at sexually antagonistic alleles, then we should expect that if they're good for females and bad for males, then they should be dominant in females, because the males are going to express these alleles no matter what, since they're hemizygous. So they're going to experience the cost always. But then if it's something that's good for females, you would want the females to get the benefit every time, and not only experience a benefit when you have 2 copies of something. So we would expect that things that are female beneficial should be dominant. And for body size, we definitely saw that. So this is the wild type, and this is an individual that has 2 copies of a female-limited X chromosome. So the figures that I've shown previously, we're always looking at 2 different types of female genotypes with either 1 evolved X or 2 evolved X's. Sorry, 1 evolved X there and 2 evolved X's there. And often there was no significant difference between them, so we pooled them for simplicity. But if you do disentangle them, then you can look to see if you have some sort of signature of a dominance effect. So that an individual that has 2 evolved X chromosomes has a similar phenotype to one that only has 1 evolved X chromosome. So here we're basically looking at the sort of significance of the deviation from the additive expectation here. We also found the same for development time, that the female limited X chromosome populations, they decrease their development time, and this seems to be a more or less dominant effect because there's no difference between the heterozygous and the homozygous females for the evolved X. So that was good. But then we did get some kind of weird results. So this change in development time, which we would expect as a signature of feminization, we didn't only see it in the FLX treatment, we also saw it in the CFM treatment, that is the methodological control. So this seems to be more some sort of confounding effect of the balancer, which was unexpected, because there's no reason why we would think that the balancer would have any sort of sex-specific benefit. When we looked at sperm competition and male fitness, then we also saw that we had some sort of effect that was looking more like an effect of the balancer that was being canceled out. So here we have male fitness in the wild type, male fitness in the FLX, and they're very similar. And then it seems like the ones with the balancer that are evolved under the presence of the balancer are actually doing better. And when you look at sperm competitive ability, you see the same thing, sperm offense. So that was really strange. I presented these results at a conference, and someone, I think it was Claudia Fricke, said to me, well, but what about interactions between the sexes during mating? And this was kind of the key to figuring out what was going on here, because these males that carry the FM balancer, they have these bar eyes, and they're basically blind. And when I realized this, I thought, oh my God, I feel so stupid. How could I not think of that? But I was so focused on thinking about the FM balancer as potentially accumulating deleterious mutations that I didn't really think about how the markers would affect fitness. And when we measured this later on, then we found that male fitness was clearly more affected by the balancer because of their homozygosity. So here we have females that have 2 copies of the evolved X versus 1 copy of the evolved X and 1 FM balancer the same way as they would be in the selection populations. And for the females, there's no difference at all, but for the males, there's a really striking difference. So that the males that get an evolved X are doing fine, but the ones that have a Y chromosome and the FM balancer, i.e. the ones that are in these sort of selection populations, they do very poorly. I mean, we always remove the balancer before we measure any phenotypes so that we wouldn't be getting confounds there. So this was a bit frustrating to realize this so late in the game. On the other hand, it did open up some new avenues for trying to figure out what was going on that turned out to be kind of exciting. So it seems like inadvertently what we did here was subject different parts of the genome to different types of selection. So that if we think of the control wild type as being sort of the normal situation, then you have sort of similar selection pressures on the autosomes between males and females, and then the X is experiencing selection from both males and females. But perhaps in the CFM flies, that what is happening is that the selection pressures on the X are the same, but because these males are blind and they're doing so terribly that you might have some sort of selection on the autosomes to compensate for that. So you might end up then with actually some sort of male-specific selection on the autosomes. And then in the FLX, then what we see is we would expect that we've been successful in changing the selection pressures on the X, but that as the result of the presence of the balancer, we might actually have some sort of opposite selection on the autosomes. So this could be one of the reasons why we didn't see an awful lot of striking differences when we looked for them, because we actually had some sort of genetic tug of war going on in this treatment between the different parts of the genome. And this also seems to have implications for the intensity of sexual conflict overall, so that conflicts over mating rate are probably quite high in the wild type population, but the presence of the balancer probably has shifted control over mating to the females. So we have some behavioral data that suggests that the males basically are really easy for the female to reject, that they wander around, and then when they come in contact with the female, they try to court her, but if she's not into it, she just walks away, and then the male can't find her. He can't pursue her. So we seem to have had a major change here in the mating dynamics. So this is in one sense, unfortunate because it means that our results are less clear-cut than we had hoped, but on the other hand, kind of interesting because it means that we can potentially try to look at the interactions between these two types of forces, that is, sort of sex-specific or sexually antagonistic selection pressures on the genome, but then also these effects of sexual conflict over mating rates. And one really interesting result is that it seems to have affected the genetic architecture as a side effect. I could back up just briefly here and mention that we've actually tried to disentangle the effect of the X and the autosomes here by picking them out and then putting them into a controlled background. And we do have some evidence actually that this is the case, that if you have autosomes from CFM and FLX, then you get masculinization of locomotor activity, but you see the opposite pattern when you look at individuals that carry X chromosomes from these populations. Right, so here we did a big quant-gen assay and looked at wing length, desiccation resistance, and locomotor activity in males and in females. And we have two replicate populations that we looked at in this case. So one is above the diagonal and one is below the diagonal. And I know that this is kind of a busy figure, but what I want you to look at is particularly here that the genetic correlation between locomotor activity between the sexes seems to, in both replicates, actually have been broken down. So these are not significantly different from 0. And so here the yellow is the wild type, and the green is the CFM, and the red, again, is the FLX. And so the ellipses here are showing both the amount of genetic variation for these traits and then also the correlation between the sexes. So we clearly have a positive genetic correlation in 2 of the treatments here and a 0 in the FLX, and the same thing for the other replicate. So that, I think, is really interesting because that's been, one of these questions as well. How do you break down genetic correlations for traits? And so potentially then this sort of antagonistic selection on different parts of the genome could be one process. So wrapping up then, some of the stuff that we have got ongoing with this project, we don't have the fly lines available anymore. They're very intensive to maintain, and so after a while, when the project was starting to reach its end, then we decided not to maintain them anymore, but we still have quite a lot of data, and we have a lot of sequence data that we can work with. So even though I haven't talked about it in a lot of detail, we have some more thorough analysis of gene expression and SNP data ongoing. This experimental disentangling of the autosomes and the X chromosome, we've done that for a couple of phenotypes, but then we also have gene expression data for these genotypes that have only the autosomes or only the X from the different selection lines, and that can be really interesting, I think, to look and see if we can see evidence of feminization or masculinization of expression in the 2 parts of the genome. And we're basically saving up all of our dominance data for a big dominance paper where we want to maybe try to see if we can see any overall patterns in which types of traits show patterns of dominance versus additivity, and if these would align with our expectations as to what things should be under sex-specific selection or not. Okay, so good. It looks like I've kept the time. I was a little bit worried because I had 41 slides. But it was easy to go through the ones in the beginning because all of you are already, quite knowledgeable when it comes to sex chromosomes. So I think the stuff that we've been doing, with the flies and the worms has been really exciting because it shows that experimental evolution does have the potential to capture at least some aspects of microevolutionary change that can reflect macroevolutionary patterns. I mean, it needs to be taken with a grain of salt, but I do think that this gives us a new approach to be able to test theories about sex chromosome evolution and not only have to rely on comparative studies where there could be a lot of other changes that happen that might not be related to the phenomenon that you're interested in. Exactly. So we can then maybe do manipulative tests of at least some of the predictions that we get from comparative studies. And as well, whenever you have this type of phenotypic change and then genomic data, then we can really try to link phenotype and genotype in the molecular and the genomic perspectives here. Right, and so this has been funded by the European Research Council, among other things, and here are some of the key people who have been involved in this project: Ted Morrow, Katrine Lund Hansen, Jesper Manat, and Jingying Li. So, thank you very much.